I guess I can turn that on. Why don't I turn that down?
So the question is, did they remember to transfer the vodcast over to this room?
I hope so. They scheduled it, and wow, is that really loud? It's on the lowest volume. Did that get any better? I just look retarded. I'll be quiet. It's on the lowest volume possible.
I hear your clothes.
I can hear you.
I can put it way down here. Is that better though? No, there's no volume knob on these. Just under the thingy. Yeah, it's very creepy. What did I do last week?
I'm on the podcast. We did 5
Can I shout out to so-and-so? We did lipoproteins and cholesterol. You have cholesterol.
I have that.
Oh.
That's last week.
Oh, the private Tim Hall thing.
Oh, yeah, I have those. No, that's the week before. Which one?
That looks really fun. I don't know what you're talking about.
Reminds me of all
It looks like
Is this just one thing?
Yeah.
Looks like glycolysis.
I hate that sound.
That.
This is last week.
Thank you.
Yeah.
And then that one divorced.
And like I said, I'll send you guys
And that kid grew up in
a drive with all this stuff on it. Cool.
And then his pages will look like this.
Hey, hey. You guys remembered where it was. Good. Alright, so we're hoping this gets vodcast. Can you guys actually hear me now at a good level? Because I don't want to be too quiet for the vodcast, if there is a vodcast. Okay, so good news is this chapter is primarily summarized on this sheet you have in front of you. So this is what I made for this chapter. It has most everything we're going to be talking about, but I will teach from the lecture manual, and we'll come back and talk about this stuff a little bit as we go. So if you're listening from the vodcast, make sure you get one of these sheets, get a copy from someone. This is your summary of Chapter 7, which is the Urea Cycle. Any questions from previous stuff? And you need to vent about anything? What?
Other than the ring?
I have a
just
question.
can't stand under
Doctor's
stuff.
been going through a bunch of the specific reactions.
Yeah, from the last chapter.
He's just telling us oh, this is leading hydrogen. We don't even know anything actual pathways for something.
No.
Just enzymes.
Mm-hmm. Not unless he's changing the format.
Okay.
You're talking about Chapter 6, right?
He's still on it.
Yeah, he just had he just went over his TV.
Oh, wow, he's way behind.
Yeah, he assumes he's going to cover 3 chapters before the next step.
Yeah, that's what I was rushing to do. So you guys have the vodcast of it. No, to my knowledge, there aren't any specific reactions. You do need to know, what are the big 3 you need to know? Yep. And what type of reactions are those called? Transamination. What cofactor, coenzyme do they use? B6, aka? Pyridoxal phosphate. Good. So you need to know those. You need to know the example reactions for tetrahydrofolate and tetrahydrobiopterin. I think those are the only, and then you need to know the exact enzyme names for the pathologies of amino acid metabolism in the back of the chapter. But those are the only specific reactions you really need to know. Did you guys get a sheet? Say it louder. You're on vodcast. Okay, so we're dealing with Chapter 7. Chapter 7 is the Urea Cycle. Don't put a rib out, or an eye, or whatever else, the kidney she's got back there. So Chapter 7 is the Urea Cycle. The Urea Cycle is our nitrogenous waste excretion system. The Urea Cycle is our nitrogenous waste excretion cycle. Where do we get our nitrogen? Amino acids, which come in what macromolecule? Is that what you do all class? Which come in what macromolecule? What do amino acids build? Polypeptides, aka proteins. So we get our amino acid, or we get our amino groups from proteins. The amino groups is what we're going to be dealing with here in the nitrogenous waste cycle, because the only thing we consume nitrogen in primarily is proteins, and the only thing we need nitrogen for is to build proteins. So if you have an excess of protein consumption, or if you have more protein breakdown than protein synthesis, you're out of balance, and you have to have something to do with those nitrogenous waste products. And it's very important because the ammonia ions that we make are neurotoxic. They're very dangerous, so you need a system to get rid of this. That's what this chapter is about. So last chapter we talked about amino acid metabolism, and we said we could break our amino acids down into two skeletons. We could break them down into carbon skeletons, and we can break them down into nitrogen skeletons. The carbon skeletons go towards either being ketogenic or glucogenic, either forming glucose intermediates or ketone bodies. That's what we were talking about last chapter. What we didn't talk about at all was the nitrogen skeleton, and all the nitrogen skeleton is not the carbon skeleton. So the whole amino acid is the carbon skeleton except for the amino group. There's a couple of amino acids that are an exception, and those would be your nitrogen-containing side groups, like your histidine and stuff like that. Histidine? Yeah, your basic amino acids. So we'll be looking at those a little bit. But this chapter is about the processing and excretion of nitrogenous waste products, which when they build up are neurotoxic and dangerous. So we have to be able to deal with that. So we're on page number 75 in your lecture manual, or in your book somewhere, chapter 7, whatever you happen to be looking at in front of you. And we're going to deal with a couple basic concepts real quick. I think these are up in the top left corner of the sheet I made for you, so feel free to look at them either place, but I'm going to go through them in the lecture manual for now. Pepsin cuts proteins in the stomach. You guys have probably learned this in physio. If not, you will be going through this soon. Pepsin is the primary protein digester in the stomach. It deals with large proteins. It deals with the whole protein that's coming in. Once we get down from pepsin in the stomach, which cleaves them in big chunks, we get down to the small intestines, which I guess you really don't need to know for this class, and we deal with trypsin and chymotrypsin. Trypsin and chymotrypsin do the same thing. They cleave proteins, but they don't do it in the stomach. They do it in the small intestine. And they don't deal with quite as larger proteins as pepsin, to my knowledge. They deal with smaller proteins. So pepsin takes these huge rocks, these huge proteins, and breaks them into smaller chunks. And then you get to the small intestines, and trypsin and chymotrypsin break them down into primarily dipeptides. Dipeptides. They do not break them down into amino acids. Aminopeptidase and carboxypeptidase will take these small guys and break them even further. And what aminopeptidase and carboxypeptidase do is break amino acids off either the amino terminus or the carboxy terminus, as it says there. So pepsin breaks them down from big proteins into little proteins. Trypsin, chymotrypsin breaks them down into small proteins. And then aminopeptidase and carboxypeptidase break individual amino acids off the amino terminus or the carboxy terminus. That's why they're named aminopeptidase and carboxypeptidase. It just tells what terminus they rip them off of, what their mechanism, or where they attach on the protein. Any questions on that? Why L amino acids? Because that's all that humans use. Humans use L amino acids and D sugars. Good. L amino acids undergo oxidative catabolism under 3 circumstances. Leftover L amino acids from normal protein turnover are degraded. In other words, we have normal protein turnover. We have some excess. We have to get rid of it. Dietary L amino acids that exceeds the body's protein synthesis. If you have a high protein diet and you don't need to synthesize that much protein, you have to get rid of the excess. And proteins in the body are broken down to supply L amino acids for catabolism when carbohydrates are in short supply. Remember when we talked about in starvation and fasting, well, primarily starvation, you start breaking down your muscle proteins to supply glucose to the tissues that need it. Well, when you're breaking down your muscle proteins, you're getting this extra waste product, which you need to get rid of. So anytime you're breaking down more protein than you're using, you're going to activate your urea cycle, and that's what we're talking about this chapter. Now, please note that this is one of the reasons that people who lift weights, bodybuilders, take extra protein, because to build protein, you must have protein. And this is a key concept. We do not store amino acids. We do not store unessential proteins. It's readily available, or we get rid of it. So there's some that are floating around the digestive system and probably a few in the bloodstream, but if we want to make a massive supply of new protein, we need an influx of new protein. We don't store this stuff. We store fat, we store glucose for later, but we do not store amino acids for later, which means that we must deal with them. I wrote this in for my own purposes because I always get confused on this and still to this day do. A nitrogen with 4 things attached to it, usually those things we're talking about are going to be hydrogens, is an ammonium ion. A nitrogen with 3 things attached to it is an ammonia ion. Easiest way to remember this is ammonium is an extra letter. It's got an extra thing attached to it. Ammonium is NH4, ammonia is NH3, or whatever else it happens to be attached to. But we're talking primarily about hydrogens in this chapter. So ammonium, 4 things, ammonia, 3 things. So once amino acids are generated through protein degradation, they're used to synthesize new proteins or synthesize other important molecules in our body like fatty acids, lipids, carbs, etc., etc. We use them to build stuff that needs nitrogen in it. Surplus amino acids are degraded further for their carbon skeleton, their nitrogen skeleton. We already talked about that. Carbon skeleton goes either to gluconeogenesis or fatty acid synthesis through ketone bodies. So that's your ketogenic, glucogenic fates. The nitrogen skeleton is what chapter 7 is about. Nitrogen skeleton of amino acid, removal of the amino acid nitrogen as ammonia. How many things does ammonia have attached to it? Good. Ammonia. It's catabolized to form the ammonium ion, which is toxic. I wrote it in, you'll see it in a couple places. The ammonium ion is toxic. Cool? So, we've talked about these reactions. I'm going to hit them again real quick, and then we're going to move on. Transamination reactions. What is transamination? Transferring a what? An amino group. I wasn't sure if you said amino acid or amino group. Transaminase or aminotransferase removes the alpha amino group using pyridoxal phosphate. Got to know it. Guess what? He's going to go through the same 3 on the bottom of this page. Pyruvate to alanine, oxaloacetate to aspartate, and alpha-ketoglutarate to glutamate. Please, 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 please memorize those. Okay. We'll go back down. So, in the transamination reaction, he's got an example here. Alpha-ketoglutarate is an alpha-keto acid going to glutamate, which is an amino acid. The only difference between an alpha-keto acid and an amino acid is the presence of the amino group on that alpha carbon. The only difference between an alpha-keto acid and an amino acid is the presence of the amino group on that alpha carbon. So if we have an amino donor, we can take alpha-ketoglutarate to glutamate, and this amino acid becomes an alpha-keto acid. They're just transferring an amino group. One goes one way, one goes the other way. Again, it's in the presence of an aminotransferase or a transaminase, and it requires pyridoxal phosphate, which is abbreviated PLP, pyridoxal phosphate. The main purpose, and please star this, the main purpose of transamination is to collect the amino groups from many different amino acids in the form of L-glutamate. We collect them all on glutamate in the liver. The liver is primarily where we're going to be doing our urea cycle. This is what we've talked about before, where the body likes to deal with one currency. It's got enzymes to get the amino group off of glutamate and make urea out of it in the liver, which means that all we have to do is everything else has to converge on glutamate. We need one enzymatic process to deal with glutamate, and everything else just gets it to glutamate. So that's what we're doing at the beginning of this chapter, is we're getting all of our other amino acids to glutamate so that we can deaminate glutamate. Questions? Cool. Exception of transamination reactions, and exceptions are always good. Lysine, threonine, proline, and hydroxyproline do not participate in aminotransferase. That's all we're going to say about it. I don't know what their fate is, but lysine, proline, threonine, and hydroxyproline, so basically proline, do not participate in what we're going to be talking about. Got to know those exceptions just in case. Every other amino acid that you deal with in your body, including those that we don't necessarily talk about as the 20 human amino acids, the ones that we use to build proteins, 21. But I mean taurine and ornithine and all of those things you ingest, those are amino acids. They're just not used in our proteins. So here's those big 3 again. Pyruvate to alanine, oxaloacetate to aspartate, and alpha-ketoglutarate to glutamate. Yes, ma'am.
Does anybody else want to know how, like, his notes, like lecture 7 notes? Like the PowerPoint? Dude, none of this is on his stuff.
All right, we're back on. Had to change the battery there. I'm going to have to come up with excuses now. Battery's going to die like 3 times a session.
It's like saying, you do the little last bit.
I know, right? In your class too? Dude, he kills the battery in our class. I don't know if he's magnetic or something.
No, he stores them up, and then we'll keep reusing the very last little bit of each one. Make sure he gets all the juice.
We found out last try that he was putting the used batteries back in the battery box as opposed to just leaving them out. And it's like, you can't do that. No wonder they run out like every day. Because literally, we had our battery changing like 1 to 2 times a day. Okay. Removal of the alpha amino acid as amino acid nitrogen as ammonia. So the alpha amino group is removed from most L-amino acids, excepting these guys up here. First and collected in the liver to form glutamate. I wrote down here, like we talked about, surplus amino acids cannot be stored, which means that we must break them down and get rid of them. We store glucose, we store fat, we do not store amino acids, we do not store excess proteins. You may build proteins that you use, but you do not store excess as a source of amino acids for later. Okay, so let's go to here real quick. So this looks complicated as hell, and I'm sorry for that. There's a lot of information on here. It kind of combines stuff from a bunch of different pages. But we're going to be dealing with this primarily for the rest of the chapter. So let me get a pointy thing so people on the podcast can watch. So we're up here, we're dealing with glutamate. If you notice, this whole section between these 2 lines is the liver mitochondria. So the urea cycle happens in 2 subcellular locations. It happens in both the liver mitochondria and the liver cytosol. That is something you got to know. Most of it happens in the liver mitochondria. I don't know if he's going to test you over which parts happen in the cytosol. He traditionally has not. But you do have to know that it occurs both in the liver mitochondria and in the liver cytosol. So we start out, we've got this glutamate in the liver, in the mitochondria. That's what we're dealing with right now. It's oxidatively, deaminated. So what are we getting? We're getting the amino group off of it. This glutamate is deaminated to yield NH4. What happens to glutamate when I take its amino group off? What does it become? What? What happens when you transaminate glutamate? Alpha-ketoglutarate, because this is a transamination. We're just not, well, it's kind of a transamination. It's not a true transamination reaction, but we're doing the same thing as a transamination. We're ripping an amino group off. So glutamate will become alpha-ketoglutarate right down here, just like the transamination. This happens in the presence of NAD or NADP. TQ happens in the presence of NAD or NADP, and it produces NADH or NADPH. What uses NAD or NADP and produces NADPH or NADH? Dehydrogenase. So what are we going to call this guy? Glutamate dehydrogenase, because that's what he does. He dehydrogenases this glutamate. Now that's really the transfer of electrons, which is taking protons with it, but in the process he's also ripping off this NH4. What does that say about this process? Is it endergonic or exergonic? What does that say if we're making NADH and or NADPH about this process? Is this endergonic or exergonic? Exergonic, good, because we're trapping that release of energy as NADH, NADPH, because those are intermediates that we can then send to electron transport that can generate energy for us. So their formation is an endergonic process. Therefore, this reaction is an exergonic process. Okay. Where's he going to notes next? Okay. So that's all we're going to talk about right now on this sheet is just this first step right here, is the oxidative deamination of glutamate by glutamate dehydrogenase in the liver mitochondria. Back on your little sheets. Glutamate is oxidatively deaminated on page 78 by glutamate dehydrogenase producing an NH4 ion, an ammonium ion, and alpha-ketoglutarate in the presence of either NAD or NADP. TQ, either NAD or NADP. Doesn't care which. It's just got to have one. Most of these reactions are specific. What do you think it says about a reaction that will use whatever is around to catch electrons? It's got to happen. It's important. If it will use whatever substrate's hanging around to dump its electrons on, it means that this process must go on. If it just used NAD, what happens when you drink alcohol? You load all your NADs up as NADHs, and then this would shut down. If it just used NADPH, what happens or NADP, what happens if you went into a crisis where you used all your NADP or you lost all your NADP? Then you have a problem. Now, what's the one thing that could shut this process down? What do NAD and NADP have in common? I guess we haven't really talked about this yet. They're both NADs, which means they both come from vitamin B3, which is niacin. So we'll talk about that later on. When we talk about niacin, I forgot you guys haven't covered vitamin biochemistry yet. I haven't taught it to you. Or Dr. Starkar hasn't taught it to you. So NAD and NADP are both niacin. If you have a niacin deficiency, this is going to shut down. A lot of other things are going to shut down first, which will probably cause problems. Reaction is reversible, which my little sheet does not have on it, so you might want to draw a little arrow on there. You guys get where that arrow goes? For those of you playing the home game, that arrow might go right there. Just so you know. Within the mitochondria of most cells. So this happens in the mitochondria, et cetera, et cetera. Other amino acids that are also directly deaminated to ammonia are histidine, serine, threonine, glutamine, and asparagine, TQ. Another memorization, histidine, serine, threonine, glutamine, and asparagine. If you'll notice on this sheet, those guys are drawn in right here. Histidine, serine, threonine, glutamine, and asparagine can also be directly deaminated. So that's just when you're studying, make sure you glance at that, telling you that glutamate is the primary guy we deaminate, but these 5 other amino acids can also be directly deaminated. In other words, they just dump off their ammonia and contribute it to the urea cycle in the mitochondria of the liver. Okay. Urea is synthesized in the liver by the enzymes of the urea cycle. Sorry, at the bottom of the page. Urea is synthesized in the liver by the enzymes of the urea cycle, which we have not yet talked about. I would know this structure. I would draw that. If you do not have it drawn somewhere, it's on my sheet in the bottom left corner. I would know that structure. There will likely be a structural question, either asking for a description or asking you to identify it based on a description, or showing you pictures of a urea molecule and asking you to identify which one is the proper urea molecule. Now you're going to learn that it has 2 nitrogens on it, 2 amino groups on it. And we're going to talk about where those 2 amino groups come from and which order they come in on, what contributes the first, what contributes the second. But the key is it's 2 amino groups bound around a carbonyl. All right. Now, this is important. Top of page 79. Glutamate is virtually impermeable to the cellular membrane. What does that mean? We can't get in or out. Where does the urea cycle happen? Where do we oxidatively deaminate glutamate? Mitochondria of the liver. Which means that anywhere else we're breaking down amino acids, we have to have a method to get stuff to the liver as glutamate. So that's what we're going to talk about for the next little bit. Because we break down protein in lots of tissues. If we go into starvation, where's the damn speaker? If we break down protein, if we go into starvation, we break down proteins. What's the primary location of protein storage in our body? Your muscles. They're not real essential. If you go into starvation, you start eating your own muscles. So you're going to be breaking down protein in your muscles, which can't deal with the urea. So we need to be able to get the nitrogen from here to the liver. So that's what we're going to talk about. Because glutamate will not pass through these cellular membranes. So there are two ways we do it. There are 2 forms that are produced from glutamate to transfer the amino acid nitrogen, either alanine or glutamine. You produce alanine in muscles. You produce glutamine pretty much in every other tissue of the body. That means that alanine and glutamine must be permeable to the cellular membrane, and that's what we're going to talk about. So for the next little bit, we're going to be dealing with how we convert glutamate to alanine in the muscles, or whatever amino acid to alanine in the muscles, and how we convert whatever amino acid to glutamine in the muscles. So if we're in the muscles and we want to donate an amino group, because that's what we're dealing with, we want to donate an amino group to something to make alanine. What do we donate an amino group to? If we're in the muscles and we want to donate an amino group to something to turn it into alanine, what picks up that amino group?
This is the security department. Please be advised that the creek lot and bridge gates will close at 6 p.m. Please move your vehicle from the creek lot to the main campus at this time. Thank you.
In other words, what can we transaminate to alanine? Pyruvate, exactly. And that's what I want you guys to start thinking about. So if you were to ask you a test question, you're in the muscle, and something donates an amino group to form alanine. What is the receptor, or what picks up that amino group? Pyruvate. So think about your transaminations. That's why they're so important for this chapter. Because in the muscle, we donate an amino group from any other amino acid to alanine, to pyruvate, and it becomes alanine. And that alanine can go through the bloodstream to the liver, where alanine is converted back, or alanine donates its alanine donates its amino group to what? To form glutamate. Alanine donates its amino group to what? To form glutamate. Alpha-ketoglutarate. Good. So if you look on this sheet, up here in the top left corner, in muscles, we have a transaminase that takes any amino acid as its donor and donates the amino group to form alanine. What happens to that amino acid? It becomes an alpha-keto acid. What do we do with an alpha-keto acid? It's either glucogenic or ketogenic. We turn it into acetyl-CoA or ketone bodies, or we turn it into intermediates of glucose, burn it for energy, or send it out somewhere. Now, muscles don't ship out glucose, so that's not an option. So we transaminate a pyruvate to an alanine with any amino acid donor. That alanine goes through the bloodstream from the muscle to the liver mitochondria, where it is transaminated back to pyruvate, and alpha-ketoglutarate is transaminated to glutamate. Glutamate is glutamate dehydrogenase, producing a free ammonium ion. You guys get that? Alanine is how we get the free ammonia groups, the free amino groups from muscle to the liver. It has to go back to glutamate, because glutamate is what the liver deals with, but glutamate is impermeable to the cellular membrane, so we use alanine as an intermediate transporter. Okay, that's what this little thing at the bottom of the page shows, is that this is the alanine glutamate cycle, or glucose-alanine cycle. Oh, that's what I forgot to mention. So, down in here, the liver has pyruvate. Let's say we're in starving conditions. Let's say we're in starving conditions. Why are we shipping out protein from muscle? We need energy. So we're breaking down muscle protein. The muscle proteins are being, or the amino acids are being transaminated to alanine. Alanine's going to the liver, alanine's going to the liver and being transaminated back to pyruvate. We're in starvation conditions. What does the liver do with this pyruvate? What does the liver do with pyruvate in starvation conditions? What does the liver do in starvation conditions? The only place in the body that really does this. Gluconeogenesis. And pyruvate's a major substrate. Speak up, you guys. You know this stuff. Pyruvate's a major substrate of gluconeogenesis. So if the liver gets its hands on some pyruvate in the middle of this urea cycle, and we're in starving conditions, and we're breaking down this, or whatever the case is, however this intermediate gets to the liver, because remember, most of the intermediates of the TCA cycle can be turned into gluconeogenic substrates. So it gets its hands on this pyruvate, it turns it back into glucose, it ships it out because we're starving and the rest of the body needs glucose. So that's why it's called the glucose-alanine cycle. So down here, we're in the muscle. Pyruvate is transaminated to alanine. Alanine gets shipped to the liver. Alanine is transaminated back to pyruvate, forming glutamate. Pyruvate is turned into glucose. Two pyruvates specifically are turned into glucose. Glucose is shipped back to the muscle for energy. So that's what we do. Liver gets its hands on the gluconeogenic intermediates, ships them back, but it also gets its hands on the amino group, which is what we're going to be talking about once we get through the other cycle. So glutamate is then converted to urea. That's what we're talking about right now. Almost. Alanine is exported primarily from the muscle to the liver through the blood. So we've kind of talked about the cycle. That's what it's saying again. Pyruvate in the muscle reacts with glutamate to produce alanine and alpha-ketoglutarate. Pyruvate in the muscle reacts with glutamate to produce alanine and alpha-ketoglutarate. It doesn't have to be glutamate. It can be any amino acid donor that's trying to be deaminated. This is catalyzed by the enzyme alanine aminotransferase. Again, just normal. This enzyme is called the glutamate pyruvate transaminase. Alanine is transported to the liver via the blood. Once alanine arrives in the liver, it's transaminated to pyruvate, producing glutamate again, and then glutamate is oxidatively deaminated by glutamate dehydrogenase, producing alpha-ketoglutarate and a free ammonia ion. That's the purpose. We're just cycling these amino groups to the liver. Shows it down here again. Glutamine Any questions on alanine? Where do we make alanine? In the muscle, to get amino acids to the liver. Why do we make alanine and not glutamate? Because alanine is permeable to the cell membrane, glutamate is impermeable, so we have to get the amino group to the liver mitochondria. If it's not coming from the liver, it has to go through something that's permeable. Glutamine is generated from glutamate, or other amino acids, in other peripheral tissues. So muscle does alanine, other peripheral tissues do glutamine. And transported to the liver, glutamine synthetase catalyzes the addition of the free ammonia ion, forms glutamine. Glutamine crosses the tissue membrane, transported to the liver, et cetera, et cetera. We're going to show that on this little chart right here, on my little chart. So what's the difference between glutamate and glutamine? What do you guys remember? Glutamate and glutamine have the same number of carbons. What do they have different? Not oxygen. What's the end of the side chain of a glutamate? Carboxyl group. What's the end of the side chain of a glutamine? It's that nitrogen, the amide group at the end there, right? So the only difference between the glutamate and the glutamine is basically that extra. Ooh, do we have it on here? Let me show it. Here's your glutamate. Here's the alpha carbon up here. Down here in this end, there's the side chain. CH2, CH2, COOH, or COO in this case. Down here, the only difference is, here's your CO, and we attach an amino group to it. So that's what we're doing. We're attaching that extra amino group to a pre-existing glutamate. So glutamate's already present. We transaminate another amino acid to donate that amino group to glutamate, forming glutamine. You guys following? Okay. We transaminate another amino acid to form glutamine from glutamate. So it picks up that extra nitrogen through glutamine synthetase. Synthetase. What does synthetase imply? Uses an ATP. So glutamate to glutamine through glutamine synthetase. Glutamine is permeable to the cellular membrane. Glutamine goes to the liver and is is deaminated to produce a free ammonia ion. Now, when you deaminate glutamine to produce a free ammonia ion, what do you have left? Glutamate. Because up here we added an ammonium ion. Up here we added an ammonia ion to go from glutamate to glutamine. We get glutamine to the liver. It's deaminated to form a free ammonium ion. And what's left is glutamate. And then what happens to glutamate? Glutamate is oxidatively deaminated by glutamate dehydrogenase to form this NH4 ion. So when you break down glutamine, you actually get two ammonia groups out of it because glutamine has two ammonia groups on it. You guys follow that? What do you think breaks down glutamine? Glutaminase. Remember, when you add A's to the name of anything, it tends to just break it down. Glutaminase breaks down glutamine. It produces a free ammonia ion and a free glutamate. The glutamate then goes through its normal process. The ammonia goes through like it was just taken off a glutamate. So, in muscles, we form alanine. In other tissues, we form glutamine. Both of them will contribute to the urea cycle in the liver. Glutamine also serves as a source of amino acids in a variety of biosynthetic pathways. But once glutamine arrives in the liver, it's converted to produce glutamate and releases ammonium ions by glutaminase present in the liver mitochondria. That's that enzyme you see on the little sheet there. It's the last bullet on page number 81. It's broken back down into glutamate and produces free ammonium ions. All right. Top of page 82, ammonium is neurotoxic. Toxic ammonia is converted to less toxic urea, so we can excrete it. Notice it says less toxic, not non-toxic. So ammonia is neurotoxic. Please note that. I don't know if he'll ask about it, but it is an important concept. In the normal human adult, it is in nitrogen. The normal human adult is in nitrogen balance, which means that the amount of nitrogen ingested each day, mainly from dietary protein, is equal to the amount excreted. That's where the urea cycle comes in. If we are eating protein and we're not using it, we are excreting protein. Major nitrogenous excretory product is urea. That's what the urea cycle is about. So now we're going to deviate a little bit from this, and we're going to talk on our own. So we're on sheet We've talked about all this stuff up at the top. In muscles, we transaminate pyruvate to alanine. Alanine is shipped across cellular membranes because it's permeable to cellular membranes, to alanine in the liver, where it is transaminated by glutamate pyruvate transaminase, forming glutamate. In other tissues, we turn glutamate into glutamine with glutamine synthetase. Glutamine is shipped to the liver because it's permeable, where it's deaminated or broken apart by glutaminase, which produces free ammonia and glutamate. And then glutamate is oxidatively deaminated to form free ammonium and alpha-ketoglutarate, because anytime you rip the amino group off of an amino acid, it becomes an alpha-keto acid. Over here, we talked about this a little. I just want to point it out. Large proteins are cut by pepsin in the stomach and trypsin or chymotrypsin in the small intestine into peptides, smaller. And then aminopeptidase or carboxypeptidase ripping apart into L-amino acids. Okay. Now we're going to deal with the meat of this chapter, which is the urea cycle, which starts from there. I'm not going to leave it up here, but it starts at that first NH4, really. Yeah. That's what we're going to call the urea cycle, basically. Everything else has been getting ammonium into the liver. Now we have to deal with that ammonium in the liver. So what we're going to deal with is we're going to take that ammonium and we're going to build it into the first substrate. The first substrate we build is carbamoyl. I think that's spelled wrong. It's M-O-Y-L. Carbamoyl phosphate. Carbamoyl phosphate. The enzyme that makes carbamoyl phosphate is carbamoyl phosphate synthetase. Carbamoyl phosphate synthetase. And takes an ammonium ion, a bicarb, and a couple of ATPs as phosphate or as energy donors, and it makes carbamoyl phosphate. Now please note, and it's not really important, but just if you're curious where the phosphate came from, it takes 2 ATP and ditches 2 ADP and 1 PI. So it's keeping one of them. It cares about the phosphate from one, it cares about the energy from both. Carbamoyl phosphate synthetase 1 is the rate-limiting enzyme for the entire urea cycle, which means it's very important to know. It's also an enzyme that we're going to talk about in Chapter 9. There is a carbamoyl phosphate synthetase 2. This is carbamoyl phosphate synthetase 1. It is AKA mitochondrial carbamoyl phosphate synthetase. Carbamoyl phosphate synthetase 1, CPS 1, is AKA mitochondrial carbamoyl phosphate synthetase, because the next one we're going to learn is not mitochondrial. So it's important to know. It also tells you where it is. So where is this happening? Mitochondria of the liver. Fantastic. So we make our carbamoyl phosphate by combining this ammonium ion with bicarb and some energy and phosphate. We get carbamoyl phosphate. Now please note that this ammonium ion donates the first nitrogen of urea. That's important to know. This ammonium ion donates the first nitrogen of urea. You'd have to know where the second one comes from too. That's the rate-limiting enzyme. We'll come back and we'll talk about the activation and inhibition of it later, but right now we're just dealing with the main pathway. So carbamoyl phosphate synthetase 1 makes carbamoyl phosphate. Now this is going to look a hell of a lot like the TCA cycle. Carbamoyl phosphate combines with ornithine. Ornithine and carbamoyl phosphate equals citrulline. They even have the same letters right now. Remember, oxaloacetate, citrate, that's going to help you remember it, right? Because you know the TCA cycle. Oxaloacetate plus acetyl-CoA equals citrate. In this case, ornithine plus carbamoyl phosphate equals citrulline. Ornithine plus carbamoyl phosphate equals citrulline. That enzyme is a cruddy name. It's ornithine transcarbamoylase. Ornithine transcarbamoylase. What's it doing? It's transferring a carbamoyl, and it's an ase because it's an enzyme. Ornithine transcarbamoylase. Now this is important because now we need to go from the liver mitochondria to the liver cytosol, because we don't want to generate urea inside the mitochondria. The mitochondria are very sensitive, and they have special needs, and we don't want to produce a bunch of wasteful products in the mitochondria, especially of one of our most active organs. So we're going to get it out of the mitochondria. We've done what we need to do in the mitochondria, which is get everything there, deaminate the glutamate to get the free ammonium ion, and then turn it into carbamoyl phosphate. So we convert it to citrulline, and then we transport it with a citrulline transporter from mitochondria to cytosol, which is why the urea cycle occurs in both the mitochondria and the cytosol of the liver. So citrulline is transported to the cytosol of the liver, where it is turned into argininosuccinate. Please note it is argininosuccinate. There's an extra little ino in there. Too many letters. Not arginine succinate, not something else succinate, argininosuccinate. This happens by argininosuccinate synthetase. Argininosuccinate synthetase. The nice thing about the enzymes in this chapter is they make a lot of sense. Learn your substrates, your enzymes will flow naturally. Citrulline is turned into argininosuccinate by argininosuccinate synthetase. Why do we name something for its product instead of its substrate? Why would we name an enzyme for its product instead of its substrate? So what do we normally name enzymes for? Substrate and what they do. Why do we name it for products? Because it has multiple substrates, right? If you have multiple substrates, you can't name it for its substrate. So the second substrate is out here. It's aspartate. Aspartate comes in and joins with citrulline to form argininosuccinate. Aspartate comes in and joins with citrulline to form argininosuccinate. Aspartate contains a nitrogen, and it contributes the second nitrogen of urea. Where does the first nitrogen come from? The free ammonium, which is deaminated off of glutamate. The second nitrogen comes from aspartate over here. Now it's it's argininosuccinate synthetase, which means it needs ATP. So it's got ATP floating out here too. All three of these guys combined, they give you argininosuccinate. Argininosuccinate, am I losing anyone yet? Argininosuccinate is cleaved by argininosuccinase, because add an A's to it, it probably cleaves it, argininosuccinase, to form arginine and fumarate. Argininosuccinate is cleaved to form arginine and fumarate. We're going to deal with this bottom part later. We'll come back to it. Right now we're just going through the pathway. So argininosuccinate is cleaved by argininosuccinase to form arginine and fumarate. And then arginine is cleaved by arginase. Again, terrible, terrible enzyme names. First time you guys have ever had that, so it's cool. Arginine is cleaved by arginase to form ornithine and urea, which means that the enzyme that forms urea is arginase. The enzyme that actually physically forms urea in the urea cycle is arginase, and it's from the breakdown of arginine. Ornithine is reformed. Ornithine gets transported back into the liver mitochondria to cycle again. So it's just like the TCA cycle. We want our guys back in and back out. All we're doing is transporting the ammonium ions out of the liver so that we can, out of the other tissues, so that we can get them out of the body. So ornithine is transported back in by an ornithine transporter, and then it can go through this cycle again. And here's your structure of urea. Urea is free, it's happy, it's good to go. Where do we excrete urea? In the urine. Fantastic. You guys are smart. It goes in the urine. Some of it goes into the blood and is filtered. Some of it is excreted directly into the urine. You'll talk about all that in physio. So, a couple of things. Over here, you increase urea. High protein diet, starvation, and fasting. Why a high protein diet? Do we increase urea? You don't store amino acids, you don't store proteins, which means you've got an excess, you have a positive nitrogen balance, which means you need to get rid of it. Starvation and fasting because you're breaking it down not for amino acids, but for glucose. Your tissues need glucose, so we break down proteins to get gluconeogenic substrates. That's the process that we've talked about so far. Now, one thing you do need to know, down here, this bottom part is called the aspartate argininosuccinate shunt. The aspartate argininosuccinate shunt. You guys recognize fumarate. It's one of the last substrates in the TCA cycle, which means that if we're in the liver cytosol, and this isn't quite going to, this will probably occur in the cytosol, I don't think he says anything else, but I'll check it in the notes just to make sure. We have the enzymes present in our body, not so much in the cytosol, because we studied TCA cycles in the mitochondria. We have the enzymes to convert fumarate to malate, just like we did in the TCA cycle. From malate, we go to oxaloacetate, just like we did in the TCA cycle. And what can oxaloacetate be transaminated to? Aspartate. Which means that we get our aspartate back, it donates the second nitrogen again. So this whole process is a cyclical process. This is why you need to know your transaminations, because you can transaminate any other amino acid and donate that to oxaloacetate. It becomes aspartate. Aspartate contributes the second nitrogen of urea to the urea cycle. Questions? Basic process done. Skim through the book, and then we'll come back and talk about some activation inhibition. On the bottom of page number 82, reaction consists of 5 enzyme catalyzed steps in which the carbon of bicarb and nitrogenous NH3 and aspartate are combined to yield urea. So we talked about that. But down here, synthesis of carbamoyl phosphate, the first nitrogen of urea comes from ammonium. First nitrogen of urea comes from ammonium. Mitochondrial carbamoyl phosphate synthetase 1, mitochondrial Cps One, combines NH4, bicarb, and forms carbamoyl phosphate. This requires 2 ATP molecules. I would know that. It requires 2 ATP molecules. It's written on your chart, but make sure you know it. Cps One is the rate regulatory enzyme, the rate limiting enzyme of the urea cycle. I wrote in these are coming up later, so don't worry about that. They're on your sheet. Then we synthesize citrulline by ornithine transcarbamoylase, that weird enzyme, ornithine transcarbamoylase, which takes carbamoyl phosphate and ornithine and makes citrulline. Citrulline is transported by a citrulline transporter, and then it's turned into argininosuccinate by the addition of aspartate, which donates the second nitrogen. Argininosuccinate synthetase condenses citrulline with aspartate, forming argininosuccinate. Second nitrogen of urea requires the third molecule. Did I write that on here? Okay. I didn't write those on here, but those are important. There are 3 ATP consumed in this process. There are 3 ATP consumed in the process of forming urea. The first 2 ATP are used by CPS1, the rate regulatory enzyme. The first 2 ATP are used in the first step, CPS1. The third ATP is used where you get the second nitrogen. Same step that gives you the second nitrogen of urea gives you the third, it consumes the third ATP of the urea cycle. So make sure you know that. Third ATP, second nitrogen. So, third molecule of ATP transfers, yada, yada, yada. Don't know the details. Argininosuccinase catalyzes the next reaction, resulting in the formation of arginine. Arginine. I don't know what your notes say. Arginate? Okay, it should be arginine, I believe. Yep, it's arginine. Catalyzes the formation of arginine and fumarate from argininosuccinate. That's argininosuccinase, because remember the A's enzymes predominantly rule the urea cycle. Fumarate can be transformed back to aspartate through the argininosuccinate shunt, or aspartate argininosuccinate shunt. We talked about that at the bottom of the page. Please know the shunt. Fumarate to malate to oxaloacetate, followed by a transamination from oxaloacetate to aspartate. That's your transamination. And then you cleave arginine to ornithine and urea. That's catalyzed by arginase, forming free urea. Cool? Easy. Now a couple of deficiency states. Those are all on your sheet, so we'll go through them here. Cps One Deficiency. What happens if you have a Cps One deficiency? What can you not make? What? You can't make CP, right? You can't make carbamoyl phosphate. If you can't make carbamoyl phosphate, you can't get rid of ammonium. What's ammonia? Is it good or bad? What is it? It's a neurotoxin. So, CPS1 deficiency. Ammonia is not metabolized, leads to neurotoxic effects. Bad. If you have a CPS1 deficiency, you can't do anything with the deaminated NH4. It's still going to make it there, but you can't do anything with it. So you get a neurotoxic effect. Same thing with ornithine transcarbamoylase. It's an X-linked gene. It's similar to the neurotoxic effects seen in CPS1. Again, you can't get rid of it. Get the effects. Make sense? Easy enough? This one's even better. Argininosuccinate synthetase deficiency leads to citrullinemia. You can't break down citrulline. What do you get? Citrullinemia, which is high citrulline in the blood. So, you can't break down citrulline with argininosuccinate synthetase, or you can't combine it with something to turn it into something. You get citrulline in the blood. No big deal. And argininosuccinase deficiency leads to argininosuccinate aciduria. If you don't have argininosuccinase, you get a buildup of argininosuccinate, which doesn't go into the blood this time, but it goes into the urine, forming aciduria, which means it is an acidic substrate. So basically, first two, you get a buildup of ammonia. What's ammonia? Neurotoxic. Second two, you get a buildup of the thing it acts on, the substrate. Citrullinemia and argininosuccinate aciduria. Those are all at the bottom of the page. I'm not going to read them to you again because we just went over them, but at the bottom of page number 84, you have all 4 of the states there on your sheet. He could ask you about them, but if you know the process, if you know your substrates, you know the order, you could draw this thing out, you know what it comes, and you can take a good guess at it. If you don't have the thing that breaks down citrulline, you're going to end up with high citrulline. All right, here's a chart. This is basically the same chart you have in my sheet, except it doesn't have the structures. So it's up to you if you want the structures. You can use this one on page number 85. I would know that this is not drawn in, but you have a line going across right here in the middle of the page that separates the cytosol from the mitochondria. It even kind of says it over here, mitochondrial matrix cytosol, which means that the urea cycle occurs in both the mitochondrial matrix and the cytosol of the liver. Thank you. Now, this came up, and you're not supposed to know substrates. You're not supposed to know structures. You don't have to know structures. You do have to know substrates. Do not take that at word. You do not have to know structures. But one of the questions on my test that I missed asked about a tricarboxylic substrate. What does that mean? It means a substrate that has 3 carboxylic acids on it, which sucks because I didn't know the structures. So I'm going to point out right now that there is a structure that has 3 carboxylic acids on it, right here. Just in case it comes up again, I don't know if it will, I don't think it will, maybe it will, I have no idea. But arginine succinate has 3 carboxylic acids on here, so you could call it a tricarboxylic intermediate. Tricarboxylic intermediate. Again, don't know if it'll come up, but the rest of these guys are pretty much all dicarboxyls. Okay. On page number 86, we have the aspartate argininosuccinate shunt. Please highlight that name. That will be a TQ somewhere. Aspartate argininosuccinate shunt. And what it's doing is, here's your urea cycle. Sorry, I got to keep pointing at this. Here's your urea cycle right here. It's taking argininosuccinate and it's breaking it into arginine and fumarate. Fumarate's converted to malate. Malate goes back to the citric acid cycle. It's converted to oxaloacetate, where it's transaminated to aspartate. Aspartate comes back and does it again. That's all it's saying. We break that guy down. We've stolen the amino group off of aspartate because aspartate contributes what nitrogen? Aspartate contributes what nitrogen? Second. Who said first? I'm going to shoot you. Kick him. Aspartate contributes the second nitrogen of urea. The first nitrogen comes from ammonium. You guys are really into this today. I'm excited. All right. Now, lastly, regulation of the urea cycle. This is the last page. So be excited, because then you guys get to go today. Unless you don't want to. Couple ways we regulate it. Substrate availability. If you have lots of a substrate, what does the pathway do? Activate. So if you have a high protein diet, you turn on the urea cycle. So higher rate of ammonia production will increase the rate of urea production. So a high protein diet turns on the rate regulatory enzyme. What's the rate regulatory enzyme? Cps One, mitochondrial carbamoyl phosphate synthetase. Or allosteric activation of carbamoyl phosphate synthetase 1, allosteric activation of it. Remember, that's how we regulate our regulatory enzymes, is we allosterically activate them. This guy is allosterically activated by N-acetyl glutamate, NAG. If it gets nagged, it does something about it. If it gets nagged, it turns on. NAG allosterically activates CPS1, which increases its activity. Now, this is important. NAG synthesis is stimulated by arginine. So if you have lots of arginine, you make more NAG. If you make more NAG, you make more urea. So, N-acetyl glutamate, which could also be a byproduct, it's a glutamate derivative. It could build up when you have lots of glutamate. I don't know that, I'm guessing based on the name. But NAG synthesis is stimulated by arginine production. If arginine is produced, you make NAG. If NAG is produced, it turns on CPS1, activating your urea cycle, producing more urea. There you go. High arginine levels in the liver stimulate 2 important reactions: increased synthesis of NAG and increased synthesis of ornithine via the arginase reaction. If you have more arginine, it gets broken down into ornithine. It turns on NAG synthesis. Those guys are all right over here on your sheet. Rate-limiting enzyme is activated by increased ammonium, or ammonia in that case, increased ammonia, which is what? A high-protein diet. It's activated by NAG, which is an allosteric activator. It's activated by arginine, because arginine activates NAG synthesis, and NAG activates Cps One. All on the right side of your sheet. And lastly, on the last page, we've already talked about them, so I'm not really going to go through them. Induction of the urea cycle enzymes, a high-protein diet. More amino acids are degraded, urea production is increased. Fasting and starvation, amino acid degradation, protein degradation yields amino skeletons, carbon skeletons, nitrogen skeletons. Nitrogen skeletons are shipped to the liver via these different pathways, and voila, you have increased urea. So, if you're breaking down proteins in muscle, what is transaminated to form the thing that's shipped to the liver? Pyruvate's transaminated, forming alanine is shipped to the liver because it's permeable. What if you're in other tissues? Glutamate to glutamine. And then when glutamine is broken down in the liver, what breaks it down? Glutaminase. Remember, adenase to it. There's only like 2 enzymes you have to learn otherwise. Adenase to it. So glutaminase breaks it down. It gets it into a free ammonium and glutamate. And then glutamate is deaminated by what enzyme? Glutamate dehydrogenase, which requires what? NAD or NADP, because it doesn't care, because it's an important process. That ammonium is the what nitrogen of urea? First nitrogen. How many nitrogens are in urea? What else is in urea? 2. 2 nitrogens in urea. What else is in urea? 2 nitrogens and a C double bond O, aka a carbonyl. Good. That first ammonium ion combines with bicarb and how many ATP? 2 ATP to form carbomoyl phosphate. Bicarbomoyl phosphate synthetase 1, which is the what enzyme? Rate-limiting, rate-regulatory enzyme, which combines with what to form citrulline? Combines with ornithine by, what is that? Ornithine transcarbamylase. Weird enzyme name. And then that's converted, yada, yada, yada. I'm not going to repeat it all to you guys, because you guys are probably bored as hell. But thank you for being here. If you have any questions, come up and see me. We will be covering Chapter 8 next week, which will take you through the end of test number 2's material. Stay up on this stuff. These chapters all kind of run together. I mean, connect together, I should say, not run together. Amino acid metabolism into urea cycle and all that stuff. So, like I said, any questions? Otherwise, get out of here. Enjoy an early night, and thanks for putting up with me talking fast last week. Yeah, no worries.